Welcome back, botanists. It's me, Stephanie. This week, the plants we're talking about have something in common with you, something more in common with you. They live on land. We're going to be talking about the Bryophytes. These are the first land plants. And by first, I really mean the first to evolve onto land. Here are our learning goals. First, we will know the position of embryophytes and more specifically bryophytes within the entire plant phylogeny. We'll be able to explain and diagram the steps needed to evolve from a freshwater alga with haplontic sex to a bryophyte with diplohaplontic sex and an embryo. We'll define embryo. We'll know the short list of characters which in combination identify the bryophyte clade or the bryophyte group. Given the characteristics of the aquatic bryophyte ancestor, be able to explain the likely reason for why gametophyte generation dominates in bryophytes. And lastly, know how bryophytes, as represented by mosses, sexually reproduce. As well, know basic moss structure. Cool. Here's your vocab. As always, feel free to pause it here. Copy these down. Even if you've sort of printed out your sheet or maybe you've got it up on PowerPoint, it really helps to just have these words written down. It does something with our brain when you actually write them down, so I recommend that. And that last part is just your reading. If you'd like to reference any of these terms, I'm pretty sure all of these should be defined pretty clearly in chapter 16 in really the 6th, 7th, or 8th edition of the textbook. Okay. So the first land plants sort of evolved out of the water and onto land sometime around 490 to 440 million years ago at MYA. This was during the Ordovician period, and during this period there was a lot of moisture in the air. It was very warm in the air, lots of shallow continental seas. And so plants sort of had this opportunity to go onto land. Their sperm could potentially still swim if they needed to, because these plants that were evolving on land were very low to the ground. But something else that made them special is that they had this really hard spore coat around the spores, specifically the myospores. And this compound, there's the word. This compound is called sporopollenin. This is what makes those spores really hard. It'll be on the slide in a sec. And the sporopollenin would be sort of the stuff that makes this spore really hard. Sorry, that black didn't really work so well. And these spores would be found inside of one of these structures. So we're going to talk about all of these terms. So I've been throwing around these terms a lot. I wanted to remind us of the video that Frank did a while ago on classification and systematics. And remember that these terms, kingdom, phylum, class, order, family, genus, species, these terms are useful for communication, but they're very outdated. But we really are going to be talking about a couple of real phyla, and by real, I mean they're monophyletic groups. They're good monophyletic groups that include a common ancestor and all of its descendants. Also, a species is a species, and then a genus. These are also really sort of terms that I'm sure we'll be talking about more today and as the semester goes on, less so the others. And just to remind you, these terms are sort of nested inside of one another. So, for example, maybe here and here, maybe these are two species. S here is just for species, some random species name. Well, together, maybe those three are a genus. And then we've got like, oh, another genus there. And, oh, well, these genera make a family. And maybe, oh, there's another family right there. And, I don't know, maybe these family make, oh, I don't know, an order. And, oh, I don't know, maybe all of the, maybe all of these, including that too, make a phylum. I'm just wanted to kind of use this random cladogram with random kind of terms here to show you that all of these terms, all these traditional classifications are useful because they show nestedness. They show that we've got groups within groups. That's implying relatedness. Okay, so the species are more closely related to each other. Right, these species are more closely related to each other than species of some other genus over here. Okay, cool. Moving on. Okay, oops. So now we've got another cladogram, one of our many famous cladograms. Cyanophora, remember this was just the genus name actually for a member of Glaucophyta, that phylum, the Glaucophytes. And I just wanted to sort of throw a few things on here. We know that that term green algae, just green algae, it's paraphyletic. Notice here we've got two groups labeled green algae, but if we look at their most recent common ancestor, which is right here, we're leaving out some descendants, namely, the embryophytes, all of these. And so that term green algae is sort of not that great for us in terms of, it's not a good monophyletic group. Okay. Anyway, we know that the Charophytes, which actually Charophyta and is a monophyletic group, but the Charophytes, when I use that ending ites, Y-T-E-S, you see that in a few places on the screen here, Charophytes, Chlorophytes, Bryophytes. That is sort of a informal ending, and it doesn't necessarily imply a phylum or a kingdom or anything, really. It doesn't imply that the group is even monophyletic. It's an informal group of terms. Now, sometimes words that end, sometimes like I'm going to throw around a term that ends in ites, like Bryophytes, but actually the thing I'm talking about is monophyletic, so I'll try to make that clear. So anyway, we know that Charophytes, this group within the green algae, a group of green algae, I should say. We know that these Charophytes are more closely related to land plants for a couple of reasons. One of those main reasons, I'm going to kind of mark it right here because it's a trait that is shared between the Charophytes and all of the land plants. This is the fact that their egg, which is that female gamete, it's retained. It's retained on the haploid gametophyte stage. So I'm going to write that down, and I'm going to remind you exactly what that means here as we review life cycles. But first, let's write that. Retention of egg. Remember the egg. This is that haploid, one of those haploid gametes, the other one being a sperm. And it's retained, namely, on the gametophyte. And then what happens is, well, fertilization takes place right there in the egg where it normally takes place. It's sort of the sperm and the egg meet, but that happens on the gametophyte since the egg is retained. And so retention of egg, it sort of leads to sort of also a retention of the zygote, which is that very first, remember, it's the very first diploid structure that's formed after fertilization. Cool. That is shared between the Charophytes and all the land plants. Awesome. Well, something else crazy happens after the Charophyte. That zygote, which is retained in the gametophyte, and that haploid gametophyte, that's to back up, to remind you, that gametophyte, which is multicellular, that zygote, which would have just normally gone through meiosis right away in the Charophytes, it evolves in the Bryophytes and beyond to go through mitosis first, that zygote. And when the zygote goes through mitosis, in other words, we have a multicellular diploid structure. Cool. So in other words, we have a young sporophyte. And we call that young sporophyte an embryo. These are sort of a term that means sort of the first divisions after a zygote is formed, the first mitotic divisions, a clump of those cells. So let's write that. Multicellular embryo. Yeah, so really an embryo is just when the zygote has divided. Multicellular, meaning we've got lots of mitosis going on. And since, ancestrally in the Charophytes, the zygote would just go through meiosis immediately, before undergoing mitosis. So let me go to the next slide here to show you. Oops. That, yeah, normally, like I said, that zygote would just go through meiosis right away, and then it would produce, exactly 4 haploid cells. We know that's always the result of meiosis, is 4 haploid cells. Well, if you've just got this one cell, then that's what's going to happen. So, well, I think what happened over time is that, do a different color here, that zygote underwent mitosis. And now the cells that are going to undergo meiosis, well, they'll be more numerous. We'll be able to have more results, more products of meiosis, right? We don't just have 4 like we did in this ancestor. The embryophytes were able to have more myospores. So that's what these are called. We call these myospores because they're the result of meiosis. Okay. Now, in both of these, notice the green cells here represent those maternal, as they call them, cells. These are from the gametophyte. So the zygote in both of these pictures, imagine it's embedded on a gametophyte, on some gametophyte tissue. Same thing here. Okay, that's what that green is. Okay. Just want to show you a few slides of what we're getting ourselves into today. This is some bryophyte diversity for you. Bryophytes tend to be very small plants, like I said. They're close to the ground. Now, we're talking about non-vascular plants in this video. By that I mean we don't have xylem and phloem, and so we don't have a way to get water up very far, and so, like I said, plants, they really have to stay low to the ground. Ooh, here's some more. Wow. We end up with some really interesting reproductive structures on these plants that we're going to be talking about. Very exciting. And we also end up with these sort of long, hairy-like structures. Will this work here? Can I play this video? Yes. Check that out. See them move? It's like they're moving on their own. Well, mosses, which are some bryophytes, like the one just shown there, sort of moving after it got wet, they're able to absorb water very quickly. They're non-vascular, but those cells can take in water rapidly, they can store water, and what happened there is when their cells are taking in water, they're able to sort of move the whole rest of the plant. They sort of inflate with water, if you will. Okay, so here's some more bryophyte diversity, and now we're getting into the terms of the taxa that sort of you're required to know, the ones we'll be talking about today. Now, these are all monophyletic phyla in and of themselves. We have 3 phyla here. Anthocerophyta, the hornworts up here. We've got Hepatophyta, the liverworts down here, and then Bryophyta, the mosses. Now, I totally hear people pronounce these like Anthocerophyta and Hepatophyta and Bryophyta. That works too. Doesn't matter. Just make sure you spell it right. That's what's important, and make sure when you are pronouncing it, you sort of have all of those consonants in there. So I happen to say, I say like ophida usually for my suffixes. And here are the features that they all have in common. They all have non-vascular tissue. I already mentioned that. They do have sporopollen and walled spores, namely their myospores have that really sort of hard coating of a substance that is able to, or that allows them to sort of resist dehiscent, or resist sort of drying out. It allows them to resist harsh conditions, like really hot or really dry conditions. And so that's really useful for them being on land and all. And also they have a multicellular sporophyte, which is retained. And namely, that sporophyte, it starts off as a zygote and then it becomes an embryo. Okay, moving on. Let's start off by looking at the mosses proper. So mosses, that term moss, really they're in a monophyletic group called Bryophyta. These are plants, but they don't have true leaves and stems and roots or anything like that. Instead, what you see here is sort of like a stem-like structure, and then we have leaf-like structures, and they've got similar functions. But these leaves, they're definitely photosynthetic, but sort of the stem tissue may or may not be photosynthetic as well. They do need to be near a source of water at some point in their life. Now, they are able to really dry out for long periods of time, surprisingly long periods of time, and they're able to sort of just completely dry out in a way that they're able to sort of reabsorb something crazy like 10 or 20 times their volume from dry to or sort of empty to full. Cool. Okay, so we've got a gametophyte and a sporophyte stage. Remember, we've talked about this now a couple times, what these terms mean, but remember the sporophyte? It always, that term sporo, actually a spore doesn't imply diploid, but sporophyte implies diploid. The term sporophyte will always be a diploid structure, and the term gametophyte will always be a haploid structure. So the gametophyte will have half as many genes, if you will. And so we've got to have something must have happened, because we're right here in between those two major life stages. We're going to talk about what happened there. Notice that the gametophyte of mosses, as shown here, is very leafy-like. That's going to be as opposed to a different sort of look that we're going to see later in different bryophytes. Okay. Yeah. What else? Let's move on. Reminder of what diplohaplontic life cycles really look like. A diplohaplontic life cycle is one that, it's also known as a Spork life cycle. This is a sexual reproduction where we have meiosis and fertilization, just like the other types of sexual reproduction. But what's unique about this type is that we've got multicellular haploid stage, the gametophyte, and we have a multicellular diploid stage, the sporophyte. And that's sort of seen here, the haploid stage on top. You've got to have two gametes come together, since they both have sort of half the set of genes as a diploid individual, or a diploid cell, rather. And so two gametes will come together in fusion or fertilization, form a zygote, and the zygote will go through mitosis and form a sporophyte. That sporophyte, at least one cell somewhere on that sporophyte will, many cells usually, will undergo meiosis and form four haploid myospores. Namely, they're spores, but to be specific, we can call them myospores because they're the result of meiosis. And so that term myospore, it always implies haploid. Okay. Just like the term gametophyte implies haploid, and gamete also implies haploid. Okay, so this is a scary screen. I know, lots of terms. Let me show you which ones are important, and we're going to come back to this screen as well in a sec. Let's start up here. I showed you sort of a picture of this earlier, but we're going to come back to that in a sec. Let's start over here. Spores, namely, these are the myospores, sort of from the previous screen, because they're the result of meiosis, which just happened right there. Okay, so the two spores, the two types of spores here, they're represented by like white and gray, but really they're going to be the same color. They look identical. They're isomorphic, meaning they're the same shape, the same color. You wouldn't be able to tell them apart. But we do have two different types of spores and two different types of gametophytes. They look the same at this stage, and even this stage. But at some point, they do look different, but it's only at the very tips here that they start to look different. One is male and one is female. Okay. And the male, notice they're both of these gametophytes, the male and the female gametophytes, are both sort of leafy. The male gametophyte is going to produce sperm cells, and the female gametophyte is going to produce egg cells. That's expected. But here's some new terms. An antheridium, or antheridia, plural. Antheridium is singular. That's the structure that produces sperm on a gametophyte of all sorts of structures, including mosses. This is our first time of many times seeing the term antheridium. And similarly, that term archegonia or archegonium, this is the structure that produces the female gamete. In other words, the structure that produces eggs. Cool. Or an egg, rather. Okay. The sperm cells are all crazy looking in mosses, as you can see sort of in this picture. Okay, and then let's come back to this in a sec, because we need to talk about these, the Antheridium and the Archegonium. These are both present on the gametophyte, and so really it'd be hard to tell, maybe from a distance, but you would notice up close that some of them look sort of different on their tops, and maybe some of them like this. Maybe these are the ones that are producing sperm cells in their antheridia, and maybe the ones that sort of look like this are the ones that are producing egg cells in their archegonia. So you're not going to have a sort of a bisexual gametophyte, if you will. These will be totally separate sexes, but they might be right next to each other, like you can see here. The antheridia produce hundreds or thousands or tens of thousands of sperm cells per antheridium. And they're surrounded by, so those sperm cells are haploid. So it's the gametophyte. And the cells that are sort of surrounding those spermatogenic cells, the ones that are actually becoming sperm cells, these ones that I'm sort of highlighting in red here, these are the sterile cells. They're also haploid, but they're not going to become sperm cells. Well, the archegonium also has that sort of sterile layer of cells that's not going to become anything sexual, all haploid. And then the egg is inside of that, in the archegonium. Okay, so the archegonium is sort of the female, antheridium is sort of the male. And okay, let's see. Oh, something cool about the male gametophytes is that those, oh, actually, I know what I forgot. That term gametangium up here, that's a term that implies a gamete producing structure. So it's sort of a term that includes both antheridia and archegonia, because those are both gamete producing structures. Okay, so this gamete-producing structure, the male one, the antheridium, they have these things at the top where all of those antheridia, there's a bunch of those sort of sperm-producing stalks all clustered right next to each other, so that when a raindrop falls, it sort of splashes all of those sperm cells out, and that's what sort of is going on in this picture, and then hopefully one of those sperm cells is able to get splashed over to an area where there is an egg nearby. And that's sort of what's going on down here. Hopefully one of these little sperm cells is able to find the top part of this archegonium, which is where it needs to swim down to find the egg. Okay, so now the egg, which is on the female gametophyte, this is retained on the female gametophyte, and then fertilization takes place right there, and mitosis happens right there. So fertilization of the sperm and egg, and now we've got a zygote, and that zygote undergoes mitosis. And that's what an embryo is, really. Okay. Let me define this term for us, though, because that term embryo, it's very important. I feel like I've been throwing it around a little bit. It's a young sporophyte, right? A sporophyte implies a diploid multicellular structure. And really, it's going to be nourished by sort of some maternal tissue. So it's a young sporophyte that's embedded in the gametangium, namely the female gametangium, namely the archegonium. Embryo. Young sporophyte that is embedded in the archegonium. All right. And so from there, we've got, sort of more mitosis going on from the embryo, and then it's a full-on sort of adolescent sporophyte, and then it becomes a full-on mature sporophyte. But that mature sporophyte, right, that's right here, and then now it's right here, it is attached to the female gametophyte. Crazy. Okay, now on this slide, we can really get into those sort of nitty-gritty terms of the sporophyte in mosses. So this part down here, this leafy part, well, on all of these, actually, this represents the haploid gametophyte. So going back to this picture, oops. This represents sort of this leafy part down here. Okay. And at first, we've just got our little haploid egg at the very, very beginning. And then that egg undergoes fertilization, and it becomes a zygote. So it's kind of hard to tell what it's supposed to be in this picture, but we'll call it an egg or a zygote. Great. And now the sporophyte is undergoing mitosis here. And as it's undergoing mitosis, notice that some of this light green tissue, which is haploid gametophyte, it's sort of, let me do a different color up here. Here we go. This light green tissue, oh, that's perfect. It wraps around that young embryo that is mitotically dividing. And then at some point, that gametophyte tissue doesn't need to grow anymore, and it sort of ends up getting ripped apart right there. And then now this sporophyte, it's sort of got this little cap. It's got this little haploid cap. And the term for that little haploid cap, it's derived from the gametophyte. The term for that is the calyptra. The calyptra is that cap, and it remains on top. It's just sort of a little cap. It's just there. Cool. The very top of the sporophyte is what's going to become the sporangium. Now, the sporangium, this is a very important spot, because this is the site of meiosis. This is where meiosis is occurring and creating those sporopollenin-coated myospores we talked about earlier, which will go on to produce gametophytes, and then it all restarts. Okay. Let's, close that up. Okay, so now let's look a little bit more closely at sort of a more realistic picture of that sporangium and sporophyte. Really, the sporangium is sort of the top part of the sporophyte, if you will. And the sporangium is, like I said, it's the really exciting spot because it's the spot of the site of meiosis. And this is where cells' chromosomes are reducing from 2 copies to a single. So this is sort of the site of reduction, if you will. Okay, well, this is sort of the multiple stages of what a capsule will go through. And I'm going to use the words capsule and sporangium sort of interchangeably here. That's okay. Okay, so again, we've got that calyptra. The calyptra is haploid, and it's literally derived from that gametophyte tissue down there. And then that falls off. It's like a little sort of wizard hat type of thing, and it just falls off. There it goes. And now it exposes this underneath. This whole structure is exposed. And we've just sort of zoomed in here above the seta, which is the stalk of the sporangium that connects it to the gametophyte. Well, okay, up here at the top of this structure, after the calyptra falls off, we've got this sort of other hat. We've got like another type of hat. It's called the operculum, and it's also represented over here in this image. Oh, and by the way, the C for Calyptra, that was this right here. This is a real image of it. That's a Calyptra. Well, the Calyptra falls off, and then you got the operculum, and then the operculum falls off. Wow. And then the operculum exposes the peristome teeth. It exposes these things right here. And this is so cool down here. This is a scanning electron microscopy image of those peristome teeth. They're sort of closed like that at first, and they will sort of open and close relative to the humidity, and this will allow them to disperse their spores when the conditions are just right. So this middle picture right here, this middle diagram, is a look through the capsule as if we were to have X-ray vision. And so there's sort of these little chambers, and inside we've got a bunch of spores. Now, namely, we could be more specific and call these myospores. Again, just to be more specific if we wanted to. Okay, cool. Yeah. Here's just another look at that. How cool is that? The capsule, like I said, it'll, depending on the moisture, the humidity, those peristome teeth will either be open or closed. And this picture up here, this is actually that part that I'm sort of circling. That's the operculum that is going to fall off and expose those peristome teeth. And here we've got some that are just sort of wide open, and they've probably already released a lot of spores. Cool. So here's a little review slide of what bryophytes do. Okay. First of all, I want to say the gametophyte is dominant, right? The sporophyte is just that dingly little thing. It's on a seta, that wiry seta, the stalk. And then it's just that little capsule, the sporangium at the top. It's not doing any photosynthesis. It's just there for a pretty short period of time. The gametophyte is dominant in the mosses. Okay. And so starting up at the top here, this is just a look at their life cycle, just focusing on their terms. The capsule is where meiosis is occurring, okay? The capsule itself is part of the sporophyte tissue, and so it is diploid. And that makes sense since it's a site of meiosis. You've got to be diploid to be undergoing meiosis. It produces myospores. Those myospores will either become a female gametophyte or a male gametophyte, producing sperm or eggs respectively. But if it does produce an egg, then the egg is retained. Fertilization takes place, all of this, the egg retained, the fertilization, the zygotes all retained on the gametophyte, the female gametophyte, embryo, and even the mature sporophyte. All of this is happening on the female gametophyte. Wow. Crazy. Okay, moving on. Here's our next phylum. This phylum, Gametophyta. This is the group known as liverworts. Notice we do sometimes have structures of liverworts that are like this, where they're leafy, sort of like what we saw in the mosses. They're sort of more or less leafy looking, even though these aren't true leaves. Well, the other look of a liverwort is sort of like this one on the left, where the photosynthetic structures are just really broad and flat, and they're not separated into these stem and leaf-like structures. That's called a thallus. So here's another picture of a thallus. This is the thalloid morphology of a liverwort, and there's just two types. Liverworts are either leafy liverworts or they're thalloid liverworts. Thalloid being the adjective, sort of. Let me define that word for us here. Thallus. A thallus is really just an undifferentiated plant body. It's just some tissue that's not differentiated into leaf or stem or root-like structures. Okay, so now something else we can see in this slide here. is sort of a look through a liverwort thallus. So what I had to do to get to that point was I had to take a fine razor and take a piece of this thallus and say, like, I want to take this slice right here. I take my razor, I cut a fine slice right there, and then I take another slice right next to it, as thin as I possibly can. Way thinner than what I just drew, actually, because I've got a fine razor and I'm in the lab doing this. And then I turn that on its side, and now I'm able to see inside of it, sort of just able to see through it. So underneath, we've got these root-like structures. These are called rhizoids. It's just sort of a structure that means a root-like structure, something that's generally going to at least keep them more or less situated on their substrate. But it's not going to be doing any absorption. Not much absorption of water. Definitely no transport of water. And then in the middle, we've sort of just got a bunch of these sort of storage parenchymus cells in the middle. And then all those photosynthetic cells are at the top. And that makes sense for them to be at that very top layer. Now, something else that we find in liverworts are these pores. And these are useful, as you remember, stomata. Back in the day, learning when we talked about transpiration and plant parts, stomata were those openings of that we find in plants, right? Those were very specific types of openings where there's guard cells. Here we don't have that. We just have these sort of gaping, wide open, always open pores that are a little bit elevated relative to the rest of the surface of the thallus. But it's nice because, of course, they need gas exchange in order to have photosynthesis occurring and all of those nice photosynthetic cells, which they've got this cool thing going on where they've got this sort of layer right there, which we can see in this scanning electron image here, where they've got these elongate cells, and all of these elongate cells have chloroplasts inside of them. Cool. So liverworts do something that I just love. They do this thing called, they make these things called gemmy cups. And this is a way for them to reproduce asexually. So I know we've been talking about sexual reproduction a lot. And, you know, something I didn't really emphasize in the mosses just a few minutes ago was that, you know, all of the vegetative growth that you see of those leafy gametophytes in the mosses, that's all considered asexual growth, really. It's just sort of vegetative. It's not doing any meiosis or fertilization. Well, here we have sort of a different type of asexual growth that's less vegetative. It's more it's sort of a way for them to get little packets of cells that are hopefully maybe going to be in a good condition to withstand certain environmental conditions, or maybe it's just a way for them to sort of get out there and disperse. And these gemmy cups, they sort of act like the little splash cups in mosses, where when a rain drop lands inside of one of them, like right there, it sort of splashes these little gemmy out. And so one of these, like, ooh, that one right there, that's a gemma, or I've heard gemma, I've heard it pronounced both ways. Gemmy is plural for gemma. And then the gemmy cup would be sort of this whole structure, like one of these structures that's holding a bunch of little individual gemmy, or like this whole structure right here. Sorry, the blue. That whole thing is a gemma cup. Cool. Usually these gemmy cup just form sort of right on the surface of the thallus. Great. This is asexual. So they're literally just clones. Cool. Okay, so I've got this new set of terms, and I hope you don't dislike it that much, even though it's a really, really, really long term. Actually, there's 3 really long terms. But notice we've got this root, gamete. Well, we know gametes are haploid, gametophyte, and we also know what a gametangium is now. Remember, the gametangium was that term that meant a structure that produces gametes. Now we just have a different suffix. We have eo4s. Well, a gametangiophore is, well, it's a structure that produces gametangia. And so, or rather, it's a structure that holds gametangia, and it produces them. And really, it's these structures right here. They're sort of hard to see. They're like these little umbrella-like structures. These are gametangiophores. And then we've got some over here, too. There's a little gametangiophore. There they are. They almost look like little palm trees, but they're very, very, very tiny, usually just a centimeter or two tall. And they sort of look like little umbrellas or yeah, little palm trees. And the female gametangiophores are called archegoniophores. These are the female ones. And then the male gametangiophores are called antheridiophores. So this isn't like a totally brand new set of terms to remember because the Antheridia and the Archegonia and the Gametangia was already known to us. The EO4 is just a structure that holds those structures. Well, what are those things? These gametangiophores are found on the thallus. And so just like in mosses, we've got our myospores that sort of to the naked eye look the same. In this diagram, they've colored them, either kind of cream or gray, but to the naked eye, they look the same until they start producing their gametangiophores. So they're either going to produce an antheridiophore, there's that word, and that's how you know it's a male gametophyte, or it's going to produce an archegoniophore, and that's how you know it's a female gametophyte. Okay, well, what is an archegoniophore or an antheridiophore really? Well, this is a look through one of them. So they sort of, remember how it looked like a palm tree sort of? Well, now this is a side view looking in, like sort of into one of them as if we were to have X-ray vision or something. And they've got these little structures right here. These are the structures that produce eggs. They're the archegonia. And then the males, they also sort of had the sort of flower look to them, their little palm tree looking thing. And they have antheridia, and this is where the sperm are produced, right? And they have the same type of thing going on where the sperm will go into, will get splashed out by a raindrop, and hopefully a sperm cell will come into contact with an archegonium, which has an egg. And then really it's the same as the moss life cycle. So the egg is retained on the gametophyte. But this time, the egg, which is retained on the gametophyte, it's on the archegonia. Same thing as with the mosses. But this time the archegonium is on the archegoniophore. Right? And so we've got this zygote, which is diploid, present on the archegonial head. Okay? So we have that zygote, which is going to form a sporangia, a sporophyte. Okay? That sporophyte is found right there on the underside of the archegoniophore, because the egg was retained and the zygote was retained and the embryo was retained. And now, just like in mosses, we have a sporangium. Again, the sporangium, though, this time it's found on one of those cute little umbrella palm tree looking things after fertilization has taken place. Okay, moving on. Here's just more cool pictures of those gametangiophores. And I just wanted to be sort of clear about this sort of egg to embryo thing. We've got an egg, which is haploid, inside of an archegonium, right? And so here I'm going to circle the entire archegonium in green. Okay. The egg is that haploid part. This is the stalk right down here, just the base. The venter, let me do a different color. The venter is this part right here that's sort of enlarged. It's sort of inflated to make space sort of for the egg. This is called the venter. Okay. I don't think that's going to be in the quiz or anything. I'm just telling you. And then the neck is this elongate part. This is the part that those Only the strongest sperm cells, if you will, can sort of make it all the way down there in order to fertilize an egg and make a little diploid zygote. So that's what happened there in my little picture. So now we've got a zygote, and really remember the embryo, it's just sort of the zygote once mitosis has already started happening. It's a young sporophyte or an old zygote, if you will. It's sort of right in the middle there. Okay, now our last phylum of Bryophytes is Anthocerophyta, or Anthocerophyta. These are the hornworts, and they, like the mosses and the liverworts, they have a gametophyte dominant part of their life cycle. Their sporophyte is sort of small, sort of short-lived, but at least in this picture you can tell their sporophyte is actually photosynthetic. This part right here is the sporophyte. This is, I'm going to just say sporophyte by 2N. And then down here, all this structure, this is all the gametophyte. Yeah, that. Okay, that. Which is N. So a little bit different looking, these hornworts, than the other ones we've talked about. All right. Something that's really, really noteworthy is that they have stomata. What? Yeah, they actually have stoma. They have guard cells, and they will potentially open and close when they need to be open and closed, depending on the gas exchange between the environment and the internal environment of the plant. So the life cycle here is much sort of less complicated, actually, than the other ones. And they produce these myospores as normal. We'll start sort of there. The myospores will produce a gametophyte. This time, the gametophyte is actually just going to produce sort of one type of gametophyte. It's not going to produce a male and a female gametophyte. Instead, the Archegonium and the Antheridium will be found on the same gametophyte. So these are bisexual gametophytes. Interesting. But besides that, same story. We've got little raindrops, sperm cells, fertilizing that retained egg, the zygote embryo, also retained, of course. And now this part, I'll do this in a different color because this is important. This part up here is the sporophyte, the diploid part. And this is the site of meiosis. Actually, namely the top part here. See where it's like sort of brownish? All of these top parts. These are the sites of meiosis. And I guess, actually, in theory, maybe meiosis is starting down here. Whatever. This is where spores are getting released, at least, at the top. So what's interesting is that since the spores are getting released at the top, that's sort of where meiosis is really happening, and that's where the sporophyte is the oldest, if you will. And that's counterintuitive. Most of the things that everything that we've talked about so far in this class has an apical meristem. Apical literally means the tip of something. And here, the tip is up here, and actually that's the oldest part. Yeah, the youngest part is down here at the base of the sporophyte, but that's where it's growing. It has a basal meristem. Basal meaning sort of the base. Yeah, this has a basal meristem. Crazy. So it's like growing up like that. Wow. Okay, we'll come back to that concept in a sec. Here's a picture of the fact that they actually have stoma, and they really do open and close in almost the exact same way that we see in stoma of vascular plants. They've got these sort of microfibrils that will sort of pull them open or close, and this will be determined by environmental conditions. Now let's look more at that sporophyte. So down here at the base, like let's look at the base of like this one right here. That's what this is. And so this gametophyte tissue, the gametophyte tissue, which is all sort of this, that's this thalloid tissue at the bottom. So hornworts, they really only have that thalloid look. They don't have a leafy look at all. They're always going to have that thalloid look. And the foot of the sporophyte is always embedded in that thallus. so the foot of the sporophyte. So technically this is the beginning, or the base of the sporophyte. But this is where we've got the meristematic tissue. So we've got a basal meristem at the base. Again, this would be sort of like right here at the very base. And then as you go up, sort of since the whole thing is getting sort of pushed up, we've got our oldest cells up here. And they're sort of releasing myospores everywhere. So like one spore here, since it starts off spherical, it'll undergo meiosis and become 4 cells, and it sort of looks like a little Mercedes-Benz symbol. It looks like 3 things, but it's actually 4. The 4th is on the back side of this. We just like can't see it because this is 2-dimensional. That's what I mean by a tetrad of spores. Okay. Okay. Oh, I guess that's it. So this is just a review slide, and you can pause it. Oh, here's another study slide, and one more. This is really just a summary of everything we've been talking about. Okay, so now here we are at a nice phylogeny showing all of the taxa we've talked about today. Now, I recommend just writing this down in your own notes, literally with your hand, writing it down, or whatever, on PowerPoint, that's fine. And copy down all of the scientific names that you know so far, the botanical names, if you will. So instead of liverworts, write Gametophyta. So we hadn't really talked about this relationship between these 3 taxa earlier. But like I said, the liverworts, they're a monophyletic group in and of themselves. There's I don't know, a couple hundred or maybe a couple thousand species of liverworts. Same thing with mosses. There's many species of mosses, and they're all a monophyletic group. Same with the hornworts. Many species, they're all in a monophyletic group. But notice that these 3 groups together, the liverworts and the mosses and the hornworts, they do not form a monophyletic group. Right? They are known as the Bryophytes. We have to use that informal ending, YTES, or PHYTES, because it's a paraphyletic group. And I know that because if we look at their most recent common ancestor that all 3 of them share, it's down here. This is the most recent common ancestor that they all share, right? Because we've got older time up here, newer time up here, or more recent time up here. And we're leaving out some descendants of this common ancestor, namely everybody else that's on this lineage. So all of the vascular plants, right? So we're just grouping, if we just group these together, this is known as a paraphyletic group. Oops. Okay. So now that we got that under control, let's add a couple of things to our cladogram that we sort of know now. And maybe I'll just sort of throw some terms out that maybe you could add on your own too. Cool. Well, we talked about this. We've got sporopollenin embedded in the walls. Of spores. Yeah, cool. We've also got sporophytes present. We just have, I think that's the simplest. If I just say sporophyte, that's another way to say we have mitosis of the zygote, which is another way to say we have an embryo. Cool. I'm going to say that. Sporophyte. Awesome. That's sporophyte. To be very clear, it's retained on the gametophyte. And if you combine this with that cladogram that we talked about earlier, or that we sort of built earlier, then we already know that the gametophyte is the ancestral dominant part of the life cycle. Okay. Ancestrally, the zygote would undergo meiosis. But a sporophyte implies that the zygote underwent mitosis. Okay. Oh, something else that's shared between all 3 of these, actually, and the tracheophytes, is the presence of the Antheridium and Archegonia. So in other words, we've got gametangia present. Great. Okay. And then something else I want to add to this, and it's evidence for this close relationship of hornworts being the sister group of the Tracheophytes. Notice that hornworts is sort of closest to the vascular plants. Well, one bit of evidence for that is what we talked about earlier, the fact that hornworts have stoma. By the way, I forgot to say this. Those stoma on the hornworts, they're only present on the Sporophyte, interestingly. That's important, sorry. Those stoma, they're only present on that stalk-like structure. Interesting. Okay. Well, since we only talked about the Bryophytes today, I just want to leave you with a little bit of a spoiler alert. The next video, we're going to talk about seed-free vascular plants, the ones that are labeled SVP here. And they all have a branched sporophytes, okay? And what we're going to notice is that we have a sort of transfer of dominance from the gametophyte to the sporophyte. And that's what we're going to talk about sort of in the next video. All right, I'll see you there.